I would like to speak about the nanoscale imaging of chromatin structure and dynamic in living cells. So in general, I'm very interested to study genome organization at the entire nucleus and nanoscale resolution using optical microscope. So I would like to go just through how the techniques helped us to understand the organization of the genome. So there is two categories, if you can classify them in this way: sequencing techniques and imaging techniques. And for both techniques, we can have three classes of the organization. Just for local, let's go for nucleosomal resolution, and the intermediate that can go for okay, for the local you can go just with imaging with X-ray crystallography and electron microscope to see the structural information about the single strand of DNA and the protein bound to it. And for intermediate, that we have micro-C and also electron microscope can go in this phase. However, with global organization, we can have for the sequencing Hi-C and other technique, and for optical microscope, we have super resolution imaging. In fact, as I like this figure, super resolution microscope can go for, or optical microscope in general can go for all scales, for local and intermediate and global, because we can image the whole chromatin that can be global, and also can go for intermediate for specific domains, and even for single gene using oligopens, who can go for really for local resolution. Why I am interested to optical microscope? Because it can give us the information for both structure and dynamic. For all of these techniques can give us only one face of the coin, the structural information. However, the only technique can work with dynamic information and living cells is only the optical microscope. But optical microscope helped us to see. So back to the history, the first colorful imaging for chromosome territories came by Kremer lab using FISH technique, and then super resolution microscope came to the game in 2015. That is from Melika's lab, when they labeled the histones and using palm and storm technique in fixed cells in fibroblast and the stem cells, they could define the clutches and at nucleosomal fiber resolution. And that's just to mention here, for super resolution, really to have such image, they talk about 40,000 frames. That is take about 20 minutes. And after that, the development of oligopens that is, can go for a specific domain or tab, and even go for the whole nucleosome that came from Shawi's lab to have specific domains or specific uh, chromosomes using optical microscope. But that's only, as I said, give us a structure. For the dynamic, there is another category using optical microscope. To have the dynamic information, we need to have high signal to noise ratio for a specific spot to track it over time for time-lapse imaging. That is one example, two examples for single gene imaging that came recently, one by Bistrikis lab using this anchor system, and with cargo system using Visoka lab at Stanford. So they can really have very short label for the DNA and track it over time. At the global scale, the work came from Mashima's lab. That is, we can have single nucleosome resolution using Palm technique and single particle tracking. So we can have a global for single particle with single particle tracking. So that is kind of tracking just for the whole nucleus, just with still a lot of spots. And my developed techniques, recent one, that is DFCC, that we can have the flow fields over the whole nucleus in living cells, so we can really painting the chromosomes or chromatin based in their dynamic information. We can see here this arrows for each direction. So we can compare this one to this one. That is really for fixed cells, and that's for dynamic. I think that's the evolution or development in the field came from fixed to dynamic. What did you learn from that? We learned that is our genome is really organized in chromosomes, and these chromosomes go for compartments, either A or B compartments, and from A or B compartments, so that go for TADs until we go for this nucleosomal fiber at 11 nanometer. And from the dynamic, at the same scale, we can have just the interior movement of the histons or chromatin as highly dynamic comparing to the envelope of nuclear, comparing the nuclear envelope. And that is for single gene, that is we have a really constrained motion due to a transcription, for example. And at this stage, that there is coherent motion go for more than 2 or 3 micrometer scale over the whole nucleus. What is missing here? So what is missing really to have a techniques can link between the structure and dynamic at the same time to monitor in details about this structure, how they change, how they transit from state to state, because that is really the key to understand this dynamic structure as a transition from condensed or folded chromatin to open one to start the nuclear process. And really, I'm interested in this area from this mesoscale from 10 nanometer until 100 nanometer, because that's we can see the enclysomal fiber and you can see how it's open and close for different nuclear process. How we can merge that is what I will speak now. So to have this resolution, that is the only optical microscope can give us, that is PALM, or this single molecule localization that's presented in 2015 in fixed cells. So why PALM cannot go for this resolution? If you imagine that is we have two structures, this simulated data for two lines. If you using just with conventional PALM for single molecule localization and start to localize your molecules over time for thousands of frames, you can have this structure as two lines. However, if you apply PALM for the same time, due to the motion blur, you cannot really have high localization because the movement of molecules can give us artifact and different structure. To avoid this problem, so it is really inverse relationship between the time and the labeling density. If you have very high activation density, that we can decrease the time significantly and you can just capture the dynamic at short time. And that's exactly what we did. We use neural network that we can predict where are the molecules in very short time at high dense area. And we calculated what is the optimal resolution can get for, in our case, in chromatin labeling, for the temporal and spatial resolution, and the trade-off between the motion blur and temporal resolution give us the theoretical value about 69 nanometer plus or 5 nanometer. That is our limitation for this condition for our resolution, and that's fair enough. 360 milliseconds, that is sub-second resolution comparing to 20 or 15 minutes comparing to conventional PALM. We trained our data and simulated in different labeling density, and we got this at this condition. We applied this trained information to our experimental or our movies. We could resolve chromatin at 360 milliseconds, and we can have here just for 12 images, one super resolved image. So now we have for one video, if you have 30 millisecond acquisition time, so for 1 minute we'll have about 166 super resolution imaged in living cells. We calculated our spatial resolution. We get about 33. That is not far from the theoretical one. And from where now we have really a technique we can give us super resolution in living cells and sub-second and sub-100 nanometer resolution. As you can see here, that is just kind of selected images from our video. And you can see here, if you start from structure, you can see the change in the structure dynamic over time. To quantify this information about this, our technique, we developed segmentation method to detect the structure unit for this chromatin or for this label. We defined entity, we call it blob. We start with dimension is about 45 nanometer and length of 90 nanometer, we started to characterize the structural information of this blob by calculating the nearest neighborhood distance between each blob. We found almost it is repetitive around 100 nanometer, and we calculated the area of each blob and also the eccentricity that can give us the shape of how it looks like this entity. We call it blob because really it has elongated shape. As you can see here, that is start from 0 to 1, and 0 is spherical shape, and 1 is elliptical. Most of the of these entities were centered around 0.9 or 0.8. That's why we call it blob, because it has a elongated shape and highly dynamic movement over time. And that is, we detected this new entity using our technique over time. But now still, we get only structural information as any technique over time, but we need to quantify also the dynamic. So to quantify the dynamic, we had also some difficulties to track bulk chromatin or regular shape part of the chromatin, because normally we need for tracking single particle tracking defined spot to track it, but now we have a structure. To track this structure, we developed two different methods. The first one named DFCC, and the idea of this DFCC is built when you have two frames, like to calculate the displacement between any objective. So you have frame 1 and the frame 2, and there is time differences, that is the duration time between the frame 1 and 2, and applying optical flow technique. This technique can calculate the velocity field and or displacement field for each pixel at sub-pixel resolution. By iterating this optical flow calculation until we get this sub-pixel resolution for each two frames. After that, we have now flow field for each pixel at nanoscale resolution, but still not enough to have the quantitative information. So we interpolated this flow fields to get trajectories, and from trajectories now we have for each nucleus, for each pixel in the nucleus, one trajectory as a virtual molecule for each, as a kind of imaginary or virtual molecule for each pixel. So now we have really huge amount of data for each pixel. So we have for each nucleus about 7 or 10,000 trajectories. That is really huge amount of information, and definitely that shouldn't follow one model of motion. That's why we tested different motion model, that is for diffusion and or flow, and this, either if diffusion can be anomalous or confined, or the mix between the flow or all models. So we tested that using Bayesian statistic to see the highest probability for each model for each trajectory, and from where we can get different parameters for depending which model is highly suggested by Bayesian. So taking each model, so now we start to build a map for the dynamic information, first for the selected model, and from the selected model we can have also each characteristic like diffusion constant or alpha exponent or the length of constraint or whatever parameter we have in this model. So now we have this super resolved image using DeepBalm, and now we have this flow fields using DFCC, and also the quantitative analysis for the dynamic now came from HiD. So now we can unifying the structure and dynamic information for the same nucleus at living cells at nanoscale resolution. So to study is there is correlation between this structure and dynamic. First, we studied the autocorrelation between structure parameters and also autocorrelation between dynamic parameters. When you see here that is the nearest neighborhood distance as a characteristic for structural information, you can see here that is positively correlated. It means if you have these blobs, they are either compact together, so highly correlated when they are together. There is high, high or low, low. So high, high, it means these blobs are very close to each other, compact chromatin, or if they are open or still correlated over from 2 to 4 micrometer. And also we calculated the autocorrelation from magnitude, also give us positive correlation, but over 6 micrometer. It means the dynamic information has more memory comparing to the structure information. So went for that. After that, we studied the autocorrelation between the structure and dynamic. As you can see, there is negative correlation. Negative correlation here means when one of them is high, the second one is low. So then if you have two blobs are very close to each other, so that is low, it means they are highly dynamic. And when they are apart from each other, that is also, it means they are less dynamic. We took all of the parameters. We started to build a map about this characteristic based in the size and sorry, the area of these blobs and their dynamic. And we could see that is really chromatin structure and dynamic are really coupled uh, in their way. But this correlation doesn't mean any causation. We don't know which one influences the second one. For that, we went for another layer of analysis using the causality Gringer causality framework. We applied that for our parameter. We found that the flow magnitude as a dynamic parameter has both for both structure parameter, nearest neighborhood distance, and blob area. And from here, we discussed how can the dynamic cause the structural change. We discussed that with one of models as active semi-flexible polymer when you have a mixture between passive and active molecules. Here we can be, that is the active molecule can be different proteins like transcription factor or RNA or chromatin remodelers. And we can see now that is our kind of cartoon. We explain how our DNA structure dynamic induced change in this one and can make this blobs formation. Taking all of this information, we go for functional analysis. What I'm doing now at Suter Lab, that is to study the effect or trying to identify the factor by which Pioneer factor can shape the chromatin. So the Pioneer factor has the ability to bind to nucleosomal DNA or closed chromatin and open it to recruit another remodeler to start transcription. For this one, it's not only the structure change, so still also the dynamic change. So there is causal relationship between these parameters. That's what I'm trying to do now, to study how is the structure and dynamic change and identify the factors by which affect each other using this causality framework and the new developed method. I think my time is running, so just I will go to the last slide now. As you can see now, we could see this structure information came from the optical microscope and dynamic information. And what we added now, we have that is this blob came between as a link between the structure and dynamic studies of chromatin. To conclude, first of all, we introduced DeepBond. It is a chromatin life cell super resolution imaging that is based in deep learning. Using this technique, we identify chromatin blobs with this elongated shape with dimension of 45 nanometers and 90 nanometer. Also, we introduce high D and DXC methods to prove the dynamic of the genome in single living cells at nanoscale for bulk chromatin and over the entire nucleus. We concluded blob density correlate with the dynamic. So the chromatin density play a role for the dynamic of chromatin, and also local chromatin might be a cause impact this structural information or blob formation. And by end, I would like to thank Kirsten because I did most of the or big part of this work in her lab. And I'd like also to thank very much Roman. He was a master student at this time, and he participated in all of this work, and he did amazing work for DeepBalm and Causality, and also our collaborator Genevieve and Ben. And I would like also to thank David uh, for the ongoing work now and his support, and thank all of you for your attention.
All right, I have two questions actually. So one is regarding in comparing your blobs that you are seeing with Cavalli's lab, for example, also observe using DNA fish. So on fixed cells, he also observed a nanoscale compartment somehow. So how would you compare these results on the fixed cells with your dynamic blobs somehow? Are they the same structures or are they different?
Oh, thank you for this nice question. In fact, we have consistency with these results because we take data from Hi-C, and we applied our method for segmentation, and we take the same illumination sheet. So you take results exactly came from different labs. That's why we collaborated with Ben at MIT. He gave us his simulated chromosomes. We labeled 200 nanometer, as we see in the microscope, with fluorophore, and start to study this layer. And in fact, we get about 75 kilobase per blob. That is closely as with Cavalli's lab identified using Hi-C and imaging.
Okay. And the second question is, your results were done in a cell line. Which cell line did you use for this experiment?
This is cancer cell U2O cells, where we label the histones H2B with photoactivated protein.
And did you try with more, let's say, differentiated cells? I mean, less plastic cells. Did you try and did you see any difference?
I'm trying now with fibroblast cells, and it should take time. I expect there will be differences, yes.
In particular, I mean, I was curious in particular with the difference between euchromatin and heterochromatin. So I know that in U2Os or in cancer cells, you don't really see any big difference between the two.
In fact, we have differences between the periphery and the interior.
Yes, but for in the interior, you did not see too much effect, right? If you have interior heterochromatin versus interior euchromatin.
That is, I cannot say, because when you calculate this blob area, we average all together, but we can see gradually came from that is really this blob is higher in the periphery comparing to the interior. But definitely, if there is heterochromatin in the interior, we cannot define it without label them with particular for particular protein for heterochromatin.
Okay.
And that's very nice suggestion. I would love to make it really.
Yeah, I mean, it's very cool. And in a sense, do you have a way to correlate the compaction of the local compaction with your different dynamical behavior?
That's what we did in Hi-D. And then, yes, we compared, okay, we didn't do it with these blobs, but in our Hi-D article, we compared between the dynamic and the compaction, and we didn't find really significant correlation. So we concluded in this article, this uncoupled from the dynamic is uncoupled from the compaction.
Very nice talk. Thank you. It's a very naive question, but what kind of time scales can you image over? Can you look through a whole cell cycle, or?
In fact, we are working almost between G0 and G1.
Mm-hmm. So how long are your movies typically?
So our videos for Deep Palm it is 2 minutes, and for for Hi-D it is about 30 seconds only. Mm-hmm. Because really the competition is very heavy. If you take longer, it will take days of days. Like every video take about 24 hours.